recibir a Silvia aquí porque la conozco ya desde hace tiempo. So maybe no se llena. Pero en el micrófono, pero ya está. Bueno. Creo que está bien.
Para que se filme, para que se escuche.
Sorry, Silvia, they need me to introduce you in the microphone, so it can be done. So thank you very much for everyone to be here in the seminar. For me, it's a pleasure to present the Dr. Sylvie Reto, who is working in Paris Saclay Institute of Neuroscience. She got his master's in neuroscience in the University Pierre and Marie Curie, Paris. I hope the pronunciation was okay. She did his the PhD also at this institute. Afterwards, she went for a postdoc in developmental neuroscience in the University of San Diego, and she got the permanent position at the in Paris Saclay Institute of Neuroscience in 1995 at the Laboratory of Neurochemistry Anatomy at the University Pierre of Marie Curie, Paris. She is a group leader in developmental and evolution of the forebrain in the Institute Alfred Fersaud in Gif sur Yvette. For sure, I spelled it wrong. And she also is the research director, second in the first class in the institute. She is the head of the Department of the Developmental Evolution in the Neuroscience Institute. And for me, it's a very honor to have her here today. Thank you.
Thanks a lot, Patty. I'm really happy to be here. We had been programming this talk for a while already, and finally today it happens. We are here. So, yes, in the group we are interested in evolution of brain and behavior, and we use this blind cavefish Astyanax mexicanus model to study evolution of brain and behavior in vertebrates. So this is a vertebrate. Okay. And so today I prepared a talk on the evolution of the olfactory system in this blind cavefish Astyanax mexicanus. So first, maybe I don't need too much to introduce the system. You are probably all very familiar with the Astyanax system, but it's clearly a great model to study microevolution of adaptation to darkness. These Astyanax mexicanus cavefish live in the cave area here in Sierra Leone, north of Mexico City, that you probably all visited here. And the beauty of this system, Astyanax, is that within the same species, Astyanax mexicanus, there are several populations and several morphs, including surface fish that are just normal fish, wild-type fish, and cave fish, which have evolved in the darkness of caves. Here, a picture of the Pachon cave, and which have adapted to total and permanent darkness. And in doing so, they have undergone both gains and losses at morphological, physiological, and behavioral levels. So this is very interesting for us. Just a few things about the evolutionary history of this system. The first and very important thing is that cavefish have evolved and surfaced from, and the present-day cavefish and surface fish have evolved from surface fish-like ancestors, meaning that the ancestor of both populations living today had eyes. And the other very interesting thing is that in this region of Mexico, there are many caves hosting cavefish, which are all blind and depigmented. So here is the map of the region where the caves are located, and you see three color-coded groups of caves with the phenotype and pictures of the fish that are living in these caves. And the important thing is that at least some of these caves result from independent colonization events, making it a very good model also to study parallel or convergent evolution in the different caves. So in the group we use the blind cavefish as a natural mutant, and we compare it to the surface fish that we consider as a wild-type animal. And so here is a short description of the cavefish phenotype. The cavefish, of course, the most striking fact is that they are completely depigmented and they are eyeless. They are blind. So this is, I should say that eye loss occurs by degeneration, but that eyes first develop in embryos of cavefish. So this is interesting from a developmental point of view also. So these are the two main striking regressive traits, but we don't want to consider that the cavefish are completely degenerated animals, because they have also undergone really constructive traits, including, for example, more taste buds, more head neuromas, larger jaws, more teeth on their jaws, etc. And there are also very interesting and numerous behavioral differences, which are very interesting for us neurobiologists. Including the fact that cavefish swim in an isolated manner and very actively, whereas surface fish school and have a social behavior. About feeding behavior, cavefish feed on the bottom of the tank in aquarium, in the lab, or in the bottom of the pond in the caves in real life. And they do sort of vacuum cleaning in the bottom of the substrate with a special feeding angle at 45 degrees, which is very efficient to find food. Whereas these surface fish, they feed in the water column. I will show you in a moment 2 short videos that are really expressive about that. The cavefish have lost the aggressive behavior. At least some of the cavefish populations have. This one, which is a patchon, has lost the aggressive behavior, which is really a trademark of the surface fish counterparts, which are quite aggressive. They have a very strong hierarchical aggressiveness. The cavefish sleep very little, whereas these fish, the normal fish, they sleep normally. And they have also evolved something that has been called the vibration attraction behavior, that is mediated by neuromasts on their head, and that makes them attracted to anything that drops on the water surface in the caves, and probably helps them to find food in the dark. And so everything seems really, all these phenotypes that I just described and listed here really seem to orient us to the idea that cavefish have adapted somehow to better find food in the dark, which is of course one of the major challenges they have to cope with in their natural environment. So about the brain now, the brain phenotype of this natural mutant. Here you have dissected brains from a surface fish on the left, a cavefish on the right. And you can see right away that the brains are pretty different, at least in shapes, at macroscopic level. This is anterior and posterior. So, for example, here you can see that the olfactory bulbs of the cavefish are slightly larger than the olfactory bulbs of surface fish. The telencephalon has clearly a different shape. This is ovoid here. This is more kind of trapezoidal here. And the most striking, of course, is at the level of the optic tectum, that is the retinorecipient structure of the brain, and which is completely atrophied and hypomorphic in cavefish, whereas it is very important here in surface fish. And looking a bit more precisely at this visual system that is clearly very strongly affected in cavefish, you have here sections through the brain at the level of the eye of surface fish adult animal versus a patchon cavefish adult animal. And you can see here the very prominent part of the nervous system that is taken by the eyes in surface fish, with the very thick and important optic nerve and the optic tectum here that is really well developed, as you could see in the previous slide of the macroscopic view, compared to the patchon cavefish adult brain, where you can only find eye cysts that are remnants of the eyes that first developed during embryonic development. No optic nerve anymore. It's completely calcified and cystic, and it's covered by skin, so that the fish is really completely blind at adult stage. And here the brain, the optic tectum, you saw on the previous slide that it was completely reduced. And so, in this talk, I will discuss about the possibility of sensory compensation for the loss of visual system in the patron cavefish, in cavefish in general. And I will discuss the possibility that the olfactory system has evolved in cavefish and try to discuss the mechanisms of this evolution of this nervous system. So I need to introduce you very briefly to the olfactory system of teleosts. It is very similar, just like ours. It is composed of olfactory epithelium in the nose, with a projection through the olfactory bulbs in the brain, and then a secondary connection through posterior parts of the brain. And here in the olfactory epithelium in the nose, there are olfactory sensory neurons which express olfactory receptors which bind to odorant molecules. So for fish, the odorant molecules are diluted in water, whereas for us it's in the air, but that's the only difference. The olfactory system is also very similar to mammals with one neuron, one receptor rule. And so each of these neurons expresses one receptor and projects to one glomerulus in the olfactory bulb. So it is a very classical composition of the olfactory system, and here you have it in real on a cavefish, on a patron cavefish, 1 month old, where you can see labeled with a marker of the olfactory system, the olfactory epithelium, here with the olfactory sensory neurons, the olfactory nerve impinging on the olfactory bulb, and you can maybe distinguish the glomeruli. Okay, so at the beginning of this project in the group, we did some kind of preliminary experiment that I'd like to show you, Victor. So this is a surface fish demonstration of a chemosensory trigger of feeding behavior in Astyanax. So this is for surface fish. So you see here in the tank in the fish facility. This is very artisanal videos. It's not very exceptionally high tech, but it's very demonstrative. Here, Yannick is going to pour water in the tank, and you see that the fish continue swimming. They school, they are together, they continue swimming, nothing really happens. Ah, no, I missed the first part probably. Victor, tu peux la refaire? I talked too much and I missed the first pouring of the water, the control. So this is water. It's clean, it's water. So nothing really happens. The fish continues swimming. And then he is going to pour what we call olfactory juice, which is crushed food, crushed and filtered food pellets. And you see that it is going to trigger a very strong feeding behavior. They are searching clearly that there has been a chemosensory trigger of this active searching and foraging behavior in the water column. These fish are visual, so they will rapidly see that there is in fact nothing to eat. It was just a fake. And so they will stop searching and this excitement. Now the same demonstration, but in cavefish. Vas-y. Merci. So the cavefish, they swim here more or less in the bottom of the tank. This is water. So they continue swimming calmly and quietly in their tank. And now this is food juice that is going to come. And you will see really clearly that within a few seconds, at least only a few seconds, it triggers an excitement that is really very important. All the fish go to the bottom, and I was telling you about this very peculiar feeding behavior, this vacuum cleaning thing with the 45-degree angle. They are all in this foraging behavior within a few seconds after the chemosensory stimulation. So this was really interesting for us and suggesting that really olfaction was really something very important to feed and probably might have evolved in the dark to help these fish surviving and finding food. So here, yes, this was the original drawing by Chemin of the 45-degree feeding of the cavefish, Astyanax cavefish. So first I would like to, in the first part of the talk, I would like to show you experiments on olfactory behavior that we have performed in the lab, so in laboratory control conditions. And in the lab, we work exclusively on the Pachon cavefish population. So the Pachon cavefish is here in this red group of caves, in the Elabra group of caves, and the Pachon cave is located here. To test olfactory responses in the lab in really controlled conditions, we decided to measure responses to food-related odors using amino acids, which amino acids are food degradation products, and they are very important olfactory cues in all teleosts. And so we designed a test, which is this one here. We built rectangular boxes here with a semi-separation along the length of the box. And on one side, we are perfusing water. On the other side, we are perfusing amino acids at different concentrations. The test conditions are very controlled. The experiments are done in the dark, under infrared detection, with controlled temperature and noise, etc. We acclimatize the fish 2 hours before the test. And in the test, we put 4 one-month-old larvae, and the tests last 8 minutes. We have divided the test box in 4 quadrants, to which we have given indexes to calculate preference indexes with the position of the fish in the box. And we calculate the position of the fish and the preference index along the 8 minutes of the behavioral test. So this is how it looks. Victor, please. This is how it looks. Here you have the tracking of the 4 fish. Each fish has a color. Here is cavefish, here is surface fish with an alanine -6 molar concentration of odorant here on the right hand side. And you can see that the fish they swim all around the box during the behavioral test, and they are attracted or not to the odorant molecule. This is the type of result that we obtain. And the question is, of course, do cavefish smell better? So this is the type of result that we obtain with serine, for example, 10 to the -5 molar. And both surface fish and cavefish, surface fish are always in blue and cavefish always in red, both morphs respond with a positive attraction response to this concentration of serine. They have a beautiful response. They are attracted to this amino acid. Now, to determine whether the cavefish smell better than the surface fish, we decrease progressively the concentration of the amino acid. And here, for example, with serine, with a tenfold dilution of serine to the -6, surface fish do not respond anymore, while cavefish still respond very positively and are still attracted to serine. This is not only true for one amino acid. Here I can show you the result with alanine, which has pretty much the same pattern. Alanine to the -5 triggers a very strong attractive response in both morphs, while alanine 10 to the -6 with a tenfold dilution again doesn't trigger anything anymore in surface fish, but still has an attractive value for cavefish. So yes, the answer is yes, cavefish smell better. And the next question is, how much do they smell better? So to do that, to know that, we decrease further and further the concentration of alanine perfused in the little behavioral box. And they still respond to -9, to -10, and they stop responding at 10 to the -11 molar, which is really a very, very low concentration. So it seems that cavefish really have exceptional olfactory capabilities, are able to smell an order of 5 magnitude of difference better than their surface fish counterparts, which is relatively exceptional. And this type of detection levels, threshold of response for amino acids have been described only in sharks, to my knowledge. And an important thing also was to verify that attraction to amino acid is really olfactory driven, because there was a possibility that the gustatory system could also be involved, because the gustatory system can also trigger responses to amino acids. Although in the literature, in most teleosts, the gustatory detection threshold levels are much lower than the olfactory detection level. So, but to test and to be sure that it is really olfactory driven, we performed lesion experiments. So we perfused Triton on the olfactory epithelium of the one-month-old larvae that we use for the behavioral test. And here you see the blue thing mixed with methylene blue is Triton infusion on the left olfactory epithelium of the fish. And this leads to the complete disappearance of G-alpha immunoreactivity on the side where the fish has been lesioned, suggesting that the cilia and the microvilli of the olfactory receptors have been washed out and have been ablated, and that the fish is not going to smell anymore. And when we do that, and when we do the behavioral test on fish that have been ablated on both sides, using the high concentration of alanine that triggers the response in both surface fish and cavefish control animals, when the fish are lesioned on the two sides, they don't respond anymore, either surface fish or cavefish in red. So showing clearly that the responses that we are recording are due to the olfactory system and not the gustatory system. So now I want to, for the part 2 of the talk, I want to show you some results that we have obtained on this olfactory evolution in the wild, during field work 2 or 3 years ago now, in the Subterraneo cave. So the Subterraneo cave is located here in the Nikos area, and it's supposed to be a cave where the population is independently evolved from the Pachon cavefish that we are studying in the lab. It's a different population and it's supposed to have an independent origin. So in this Subterraneo cave, we took advantage of the fact that it is a little bit peculiar because it hosts a population of hybrid F2 generation 2 type population of cavefish. This is due to the fact that, and we have seen that 2 days ago when we were there with Louis and Patty, this is due to the fact that very regularly surface fish are washed inside the cave during the rainy season. And there they encounter cavefish and they hybridize and they breed together. And so they generate a population of fish where the characters are completely segregated. So you can find on higher power, you can find fish that are really surface fish looking, fish that are really cavefish looking, highly troglomorphic, but you can find also fish that are completely depigmented with little eyes, or fish that are a little bit pigmented with no eye at all. So the segregation of the characters is typically F2 type of population of fish. And on this type of animals, we performed the olfaction behavioral test, and we tested the chemosensory response inside the cave. So the setup was a little bit more artisanal than in the lab, of course. So we put fish in these little plastic pools, and we perfused, so this is viewed from the top, and we perfused on one side the fruit juice that I showed you in the previous videos in the lab, and on the other side, water. And we put 8 fish in the little swimming pool, 4 fish that were really troglomorphic, no eye at all, and 4 fish that were really non-troglomorphic with eyes. And here is the result of the experiment. Only the troglomorphic fish in red here, that are pictured here, were attracted by deodorant. The surface fish didn't care at all, which is a relatively surprising result, but it is a result. Only the cavefish-like and the eyeless fish were attracted to the area of the little plastic pool where the odorant was perfused. And more recently, in March of this year, we performed an experiment that was a little bit more sophisticated. With now, instead of the fruit juice, we used alanine, like in the lab, to try to see what the difference or the parallelism with what we can have as behavioral test in the lab. And we found that this subterraneo troglomorphic fish can respond to alanine 10 to the minus 9 molar, which is therefore a very low concentration of alanine, clearly a concentration that the surface fish in the lab cannot respond to. And so suggesting that there is really a parallel evolution in these two caves, Subterraneo and Pachon, where the olfactory modality is really enhanced in these troglomorphic animals. So up to now, I showed you that clearly, I hope I convinced you that cavefish really smell better than surface fish, and much better than surface fish. And we have evidence also for independent evolution of these olfactory capacities in two different and independently evolved cavefish populations: Pachon, that we have tested in the lab, and Subterraneo, that we have tested in the field. And so, of course, this asks a lot of questions, a lot of interesting questions, like these ones that I have listed here. The fact that maybe olfactory performance in cavefish could be due to the loss of eyes and loss of vision, and this is something that we have started testing in collaboration with Louis, for example. I'm not going to show you the result today. It asks also what is the neurophysiological underlying mechanism. This is really interesting from a neurophysiological point of view. And I decided that I would show you some data we have showing how did the olfactory system evolve in cavefish. So this leads me to the next part of the talk, which is about anatomy and neuroanatomy of the olfactory system. And I will stick to the peripheral level, only in the olfactory epithelium, and I will not go inside the brain in the olfactory bulbs that we have not studied yet very carefully. So here are pictures, immunofluorescence pictures of surface fish on the left, cavefish on the right, 1 month old, which have been labeled for G-alpha-olf, which is a marker of the olfactory sensory neurons. So we found, this is a Pachon cavefish always in the lab, we found that the olfactory epithelium of the Pachon cavefish is much larger. This is a high power view here of the nostril and the olfactory epithelium surface fish, cavefish. We found that the olfactory epithelium is much larger, this is quantified here, in Pachon cavefish than in surface fish. We also investigated the olfactory projection by insertion of a crystal of dye eye, that is a lipophilic dye that goes into the cell membranes and labels the axonal projections. So if you put a crystal of dye eye in the olfactory epithelium, you trace the axonal projection through the olfactory nerve and onto the glomeruli in the olfactory bulb. And we can see that it is really very similar. The olfactory nerve here in the Pachon individuals is always a bit longer, but this is probably due to the fact that the skull is larger, so it has to go a longer way to the olfactory bulb. So the global organization of this olfactory projection and system seems to be very conserved. The only difference that we could see was the size of this olfactory epithelium and nostril opening. So that was very, very interesting, this question of size. And we digged a little bit further, looking at the, I wanted to show you that, this was in Pachon in the lab, and we actually found the same thing for Subterraneo in the field, that on the fish that we had recorded for olfactory behavior. We found that the responsive animals in the Subterraneo cave, that were troglomorphic, you see there is no eye here, had larger nostrils than those that were non-responsive to the food used, a small nose and a big eye here, and this is quantified here. So apparently the cavefish have evolved large nostrils compared to surface fish. We wanted to dig a little bit further, again on the Pachon cavefish of the lab, for the olfactory sensory neurons and the composition in terms of olfactory sensory neurons that is there in the olfactory epithelium. So maybe you don't know, but I will just go very quickly through the different types of olfactory sensory neurons that are in the olfactory epithelium. So they can be recognized by morphology, by the level of the soma and nucleus in the depth of the olfactory epithelium. And they have different names. So the vast majority of olfactory sensory neurons are called ciliated neurons, 90% of them. They have specific markers, the OMP, olfactory marker protein, is specific of this type of sensory neurons. And these cells are supposed to be recognized in bile acid and alarm substance in fish where it has been tested, like surface fish or other salmonids or fish like that. The second type, the second most abundant type, is the microvillus sensory neurons. they have this appearance. They have also, it's possible to label them with specific marker that is TRPC2, it's a channel. And they are particularly sensitive to alimentary cues such as amino acids. And in the olfactory epithelium, there are also crypt cells that are fish specific, they do not exist in mammals, but it's only a very few neurons. They have their own marker as the protein S100, and they respond to sexual pheromones. And more recently, in zebrafish, a fourth population has been described, the cape neurons. It's supposed to be labeled by G. alpha, but it is a little bit controversial, this fourth population of sensory neurons. So what we did is to compare these sensory neurons with these specific markers that we have in the olfactory epithelium of cavefish and surface fish. So this is right in new results, so I'm sorry there are still some things here that are in French. I apologize for that. And let's start with the ciliated cells, the most abundant cells, labeled with OMP, which respond to bile acid and alarm substances. And so you have here these confocal images with the immunofluorescence, where you can see that really all these little green dots are neurons labeled with OMP, so they really cover all the olfactory epithelium, both in surface fish and in cavefish. And this is the merge with the DAPI labeling of the nucleus. It was clearly impossible for us to count these neurons. They are clearly much too numerous. But we have the feeling that the distribution is rather very similar in the two morphs, surface fish and cavefish. Now the second population that we looked at, the crypt cells that respond to sexual pheromones, they are much less abundant, so the images of immunofluorescence are much nicer also. So the S100 antibody labels really nicely a subpopulation of these neurons. You have the merge here on the depth of and they really cover the surface of the olfactory epithelium. And we were able to count the number of cells that was reported to the volume of olfactory epithelium, of course, and there was no difference in the distribution of these crypt cells between cavefish and surface fish. And now the interesting result came with the microvillus cells, which respond to alimentary cues, including amino acids, which are labeled with the TRPC2 antibody, which also cover very nicely the surface of the olfactory epithelium. It's a cup at this age, in 1 month old individuals. And we found that the density of TRP2 positive cells was much higher in cavefish than in surface fish per millimeter cube of volume of olfactory epithelium. So of course this was very, very interesting to us because these are the very neurons which respond to, which are supposed to respond to amino acid and alimentary cues. So the fact that they possess proportionally more of these particular olfactory sensory neurons could be an advantage for the cavefish to find food, and could maybe explain the fact that they have such high discrimination thresholds or low discrimination thresholds for very low concentrations of amino acids. So we are now performing experiments where we stimulate the fish with high or low concentrations of amino acids, and doing double labeling for activity of the neurons and for the identity of the neurons to check that these are the very neurons that really respond in Astyanax to these alimentary cues. So for this neuroanatomy for this neuroanatomy part of the talk, I showed you that cavefish have a large olfactory epithelium containing a high density of microvillus olfactory sensory neurons. And so the next question we asked was what is the developmental origin of these differences in anatomy of the olfactory epithelium in the two types of Astyanax. And so this is the fourth part of the talk about developmental neurobiology, and trying to go back to the embryonic origins of the olfactory epithelium and compare surface fish and cavefish embryos. So on this developmental staging table of Astyanax, to find the first signs of an olfactory epithelium, you need to go to this stage of embryogenesis, which is at the end of gastrulation, which is 10 hours post-fertilization. Because the olfactory epithelium, which is a peripheral sensory system, develops and derives from what is called a placode. So I don't know if you are very familiar in the audience with this type of developmental biology. So I will try to explain simply and clearly how it goes. So here you have the Astyanax embryo on the lateral view, and this is the head of the animal. And this is the skin here of the head of the animal from the dorsal view, from the top. So at the end of gastrulation, the neural plate has been formed, the neural ectoderm has been induced. And so the neural plate is here, it's the green part, where the future brain and retina are going to develop. This is really the neural ectoderm part of the embryo. And all around this neural plate, you have a horseshoe-shaped area that corresponds to the placode, that is non-neural ectoderm, and that will give rise in all vertebrates to all the sensory derivatives of the head, including the olfactory epithelium in this region here, according to phase maps, the lens of the eye, and further down, therefore posterior in the embryo, you will have the otic vesicle or the precursors of the lateral line in amphibians and teleos, for example. So all the sensory derivatives come from a placode here. And this is the placode in real, on the real Astyanax embryo, labeled with a developmental gene that is called DLX3B, but that doesn't have any importance, the name of the gene, that specifies this placodal region where the sensory organs are going to derive. And so what we did was to compare the placodal patterning between surface fish and cavefish embryos at these very early stages of development. So we used, to do that, we used in situ hybridization with specific probes for developmental genes that are known to label these different subparts of the embryo. And here is, for example, I showed PAC6, PAC6 in situ hybridization. At the level, so PAC6, as probably you know, is a very important gene for development of the eye. So it labels very strongly all the neural plate here that is going to correspond to the future retina of the animal. But it also labels these two stripes of cells here on the side of the neural plate, which are located inside the placode region, and which correspond to the future lens of the animal. So the precursor of the lens. And I hope you can see that in surface fish, the lens placode area, the lens placode presumptive domain is much larger than in cavefish, where it is really much smaller, and the anterior part is very reduced, and this is quantified here. Conversely, when we use a marker of the olfactory placode, that is called here IA2, but that, again, that has no importance, the name of the genes, and you can see that the surface fish olfactory placode is very small compared to the cavefish olfactory placode that is very much enlarged at this early stage of development, and this is quantified here. So at the end of gastrulation, in these Astyanax embryos, the cavefish lens territory is very reduced, but the olfactory territory at the level of the placode is very much enlarged. This continues during later embryonic and early larval development. You have here a marker of the lens, another marker of the lens PTX3 at about 1 day of development, where the eye is now formed with the lens in the middle. You can see that the surface fish lens is much larger than the cavefish lens. But on the contrary, using OMP, this olfactory marker protein as a marker, you can see that the olfactory organ is much larger in cavefish than in surface fish. So there are really major size differences, and from very early on in development that continue during embryonic and larval development, and that continue, that are still there in 1 month old individuals, as I showed you a couple of slides earlier. So we decided to try to investigate how, what was the underlying developmental mechanism that could explain this sort of trade-off between olfactory or visual derivatives at placodal level. And here I need to tell you one more thing at the end of gastrulation here. It is the fact that both the neural plate and the placodes developed under the influence of signaling centers that are mainly located at the midline of the embryo and that secrete morphogens and growth factor that are going to control really the growth, not only the growth, but also the patterning and the morphogenesis of the anterior, of the head, of the brain, and of the placenta. These signaling centers, as I was telling you, are located at the midline of the embryo, and there are three very important ones that we have studied. It is the notochord, the underlying notochord that secretes the morphogen sonic hedgehog that maybe you have heard of. It is relatively famous, hence its name. The anterior neural ridge here at the very tip of the neural plate secretes FGF8, it's fibroblast growth factor 8. And the tip of the notochord, that is called the polster in fish, secretes the BMP4, bone morphogenetic protein 4. And so what we are going to look at now is the influence of these three signaling centers here on the patterning of the placode and comparing always surface fish and cavefish embryos. And the very interesting thing is that these three signaling systems are modified in cavefish. The sonic hedgehog expression is much larger in surface fish than in cavefish, and this has been shown since a while now by the group of Bill Jeffrey and Yoshiyuki Yamamoto. And we have reproduced these results. Really, sonic hedgehog expression is much larger here in cavefish than in surface fish at the anterior part of the notochord at the end of gastrulation. These are all 10 hours post-fertilization embryos. We have also found that FGF8 at the anterior neural ridge, so at the very tip of the neural plate, is expressed 1.5 hours earlier in cavefish than in surface fish. So here we are in the presence of a heterochrony of expression of this morphogen. Here it was a heterotopy, it's expressed larger. Here it's a heterochrony, it's expressed earlier. And we also found differences for BMP4 that is expressed in the precaudal plate, here at the tip of the notochord. Here we have differences in the spatio-temporal regulation of the secretion of the BMP4. For example, here on this 10 HPF embryo in surface fish, it is expressed only at the very tip in what is called the polster, whereas in cavefish it is expressed in the more posterior part of the precaudal plate, but not in the polster. So this tells us that globally, the signaling environment of all these morphogenes and growth factor molecules is completely different in cavefish and surface fish embryos. And that may explain why the morphogenesis and the size of the different organs is different at the end in embryos, larvae, and later in adults. So we decided to investigate this possibility. And we used pharmacological tools to address this question. So first here I want to show you the case of sonic hedgehog, which is, I remind you, expressed more largely in cavefish than in surface fish. So we used cyclopamine signaling inhibitor, that is called cyclopamine, to reduce sonic hedgehog signaling in cavefish, therefore trying to mimic the surface fish condition. And when we do that, we find that the lens size increases and the olfactory epithelium size decreases in cavefish that have been treated with cyclopamine. This tells us that sonic hedgehog signaling inhibits lens placodal fate but stimulates lens olfactory placode fate. And therefore that hedgehog signaling has opposite effects on the two sensory fates. And since there is a difference in the level of hedgehog signaling in the two types of embryos, this could already explain the fact that cavefish have larger olfactory epithelia and smaller lenses. We did the same kind of thing with FGF signaling manipulation. We used a specific inhibitor of FGF receptor signaling that is called SU5402. It's a crazy name. And we found that when we treat cavefish embryos with this FGF signaling inhibitor, the olfactory epithelium is also reduced, like here for cyclopamine, but it doesn't affect the lens. I will show you that in a moment on the summary slide. We also wanted to address the possibility that BMP4, the difference in BMP4 expression was involved in these differences in olfactory and length derivative sizes in cavefish. But there it was not possible to use the pharmacological tools because we are not in the presence of stronger BMP4 signaling in one morph than in the other. We are in the presence of signaling in different places and really very sub-regions of the precaudal plate in the two different embryos. And so to address the question of BMP4, the impact of BMP4 signaling, we used another trick, which was injections of BMP4 protein, trying to mimic the situation of one morph in the other, as always. So we performed anterior injections of BMP4 to mimic the posterior situation in surface fish, or we performed posterior injections of BMP4 here in the posterior precaudal plate to mimic the posterior expression of BMP4 in cavefish. And I'm showing here only the results of the cavefish injections for sake of clarity. And as you can see, the anterior BMP4 injection increases the size of the lens in cavefish and decreases the size of the olfactory epithelium in these injected cavefish. The global result of the whole set of experiments, injection anterior or posterior in surface fish or in cavefish, gave the following result, that BMP4 was stimulating lens placoidal fate and inhibiting olfactory placoidal fate. So, anterior BMP4 signaling promotes lens and inhibits olfactory fate. So with all these experiments, we could start building a small regulatory network of these signaling systems and of their impact on olfactory derivatives. And if you put this in the embryonic context of the cavefish, so here is again my little scheme of the neural plate and the placode region. In cavefish, we know that sonic edgeog is increased in space of expression pattern, and this could explain the inhibitory effect on the lens fate and therefore the small lens of the cavefish, but the stimulatory effect on the olfactory fate and therefore the larger olfactory epithelium of the cavefish. We have a heterochrony of FGF8 that is expressed earlier in cavefish, and FGF8 stimulates olfactory fate, so that could also participate to the increase in olfactory epithelium size of the cavefish. And we have this BMP4 in orange that is expressed in different subparts of the precaudal plate, which also explains in part the fact that they have small lenses. So these data about really early developmental biology suggest really a trade-off between the two sensory organs, the olfactory part and the visual part. I think it's a very nice example of developmental evolution driven by this evolutionary toolbox that is in the early development. And of course, it is very tempting to suggest and to think that it may have an adaptive value for cavefish to have a big nose to smell better in the dark. And to really finish the talk, I want to, it's part 5, but it's only 2 slides, I promise. So to wrap up and to go back to behavior, from developmental biology to behavior, we have recently tested the influence of these early signaling systems on cavefish olfactory capacities, because it is of course the ultimate question. We want to wrap up and do the whole thing from developmental evolution to behavioral capacities and evolution. And so we decided to use FGF8 as a signaling molecule that was really interesting to manipulate the development of the cavefish olfactory placode, because FGF8 is the only one of these 3 signaling systems that we have looked at that only affects the olfactory placode but not the lens placode. That is maybe more specific and maybe more specific of the olfactory system. And so what we did is we treated embryos a little bit later than the end of gastrulation, but just after neurulation. We treated embryos with the FGF8, with the FGF receptor signaling inhibitor, this SU5402 that I told you about slightly earlier. And here, if you look at the cavefish at 1 day and a half of development, approximately, you can see that the treated cavefish embryos have smaller olfactory epithelia. These are confocal images cutting at the level of the head, where you have the two olfactory epithelia that are developing at the front of the head. And the effect of the treatment of the FGF signaling inhibition was quantified. And indeed, it reduces the size of the olfactory epithelium. We have tested that on 32 HPF embryos. For 5 days old embryos, the olfactory epithelium is still decreased after this very early treatment of FGF signaling inhibition. And so, of course, what we did was to test the olfactory capabilities of these treated animals using very low concentrations of alanine once they reach the juvenile stage at 3 weeks old, when they are 3 weeks old. And as you can see, the cavefish that are 3 weeks old that have been treated only with DMSO, which is the product that we use to dilute the SU compound, they respond to alanine 10 to the minus 9 molar. But the fish that have been treated with the SU compound, therefore that have shorter olfactory epithelia from the beginning of their development, do not respond anymore to this low concentration of alanine. Suggesting really that the developmental modulation of olfactory epithelium size is a critical and important parameter for future olfactory discrimination in behaving animals. So now, of course, the very interesting question is going to know whether in these few treated animals that have small epithelia, what is the situation of the olfactory sensory neurons? Do they have more of this TRP2, do they have less, or do they have the same number of these TRP2 positive olfactory sensory neurons that are supposed to be responding to these olfactory and amino acid cues? Okay, so here is a summary of the talk. I showed you that cavefish smell much better than surface fish and have a larger olfactory epithelium all their lives from the end of gastrulation to adult stage. And that seems to be an independent case of evolution, at least in 2 different cases, Pachon and Suterano. And in the second part of the talk, I showed you really early developmental sensory evolution that is triggered by morphogen controlling organ size and also probably future olfactory abilities. And I showed you also that the cavefish olfactory epithelium has a modified neuronal composition, and this we are currently trying to link these 2 last parts of the picture to really picture the global evolution of the olfactory system in Astyanax. And with this, I would like to thank the people who did the work, of course, because this is the most important. So this is a picture of a part of the group at the entrance of Subterraneo Cave a couple of years ago. And so the results I have shown today were obtained by, so the results on behavior have been obtained by Yoni Lubiovic, a previous postdoc in the lab, with Marilyn Blin, who is an engineer in the group, and Yannick Elipo, who is here, who was a previous PhD student. This is for olfactory behavior. The developmental biology part of the talk on the placard is the work of Hélène Hinault, who was a PhD, and Karen Podin, who was also a PhD in the group. And the part on the neuroanatomy and the last part on the SU treatment, I don't have the picture, but it is, I wrote his name on the slides. It was done by Eugène Tin, who is now working in Chile in olfaction in zebrafish. And I thank you for your attention, of course.
Ok, pues tenemos tiempo para un par de preguntas.
I don't know what's the time. Was it too long? Okay.
Questions? Patty.
I have a question, Sylvie, in the graph that you presented, when you measure the number of neurons related with the sexual attractions, or because I don't know if you divided or standardized the comparison between surface and cavefish in terms of the area of the...
No, it was not on the area, it was on the volume. We do confocal stacks and we do 3D reconstructions, and we use ImageG with little plugins to measure the volume of the olfactory epithelium. Because we found that the surface or the length of the fish was not a correct way to standardize and to the results. So you have really to relate this to the volume of the olfactory epithelium, because in cavefish, not only the surface is larger, but when you look on sections carefully, the depth of the olfactory epithelium is a little bit larger also. So the whole volume of the olfactory epithelium is bigger. So it was how we normalized to the volume. And I think it is important to do so.
Yeah. And in the last part of the talk, when you were just describing these networks related with the EMP4 or
the signaling system,
do you think it could be the same network also in subterranean? Because this, or at least I understand, these are only for pigeons, or it's
yes, all the work that is really it's really lab work, and this is the only on pigeon. The only thing that I showed in subterraneo was the olfactory behavior, and just the size of the nostril, but in adults. No. We have no embryos of subterraneo. We cannot do all this work on subterraneo. We only work on pigeon in the lab. So maybe there is something similar in other caves, but we don't know. The underlying embryonic mechanisms, we don't know. Bill Jeffrey had shown that, I think, that at least in several caves, the midline signaling was different. He looked at, they compared the PAC6 expression pattern in different cave population of embryos. I don't remember exactly the name, probably it was in Sabinos, Piedras, Pachon, Subterraneo, I'm almost sure not. But Tinara probably, yes, also they have. And so on their figure it was really clear that the PAC6 expression pattern, but that now at neural plate level, so the future eye, was affected more or less the same way in these different embryos of these different caves, suggesting, it's very indirect, but it suggests that the midline signaling is affected probably in a similar way in these different populations. But this is only for sonic. For FGF8 at the anterior neural ridge and for BNP at the precaudal plate, I have no idea.
Thank you.
Carlos and then Ulises.
Congratulations for your talk, and all your work is really interesting. I think that it's especially illuminating when talking about clear examples on probably selection effects. That's something I don't know.
Searching for selection marks is really going to be something very interesting. That now that we have the genome, that we can have several genomes of one population, it's going to be possible to look for selection marks. Doing selective sweep analysis, for example, that's going to be really nice. But talking about selection now, it's too early.
Yeah, probably. too early, but still I think that it could go forward that
Yeah, clearly.
Okay. So, and I had one question. Have you tried with the behavioral experiments with hybrids? And what do you think that could be the result?
Yes, we did.
Yeah.
F1, we did only with F1s, because F2s it's difficult because they are so different phenotypically. There is a big segregation of in F2. So you cannot put 4 fish that are, you don't know the fish that you are putting in the behavioral box. As we put 4 fish, we would need to have an individual test for F2. So we did not do F2s for the moment, although we are thinking about it because I think it can be very interesting. But we did only F1s, and F1s, they are very bad smellers. They smell less well than surface fish. So the genetics of this seems to be a bit complicated. That's all I can say.
Ulises.
Very interesting talk. Congratulations again.
Thank you.
I have a question about if I understood, BMP4 has different activity between cavefish and surface fish, right?
It's there, yeah, the dynamics of the expression pattern are different. You know, you're the developmental biologist in the room, so the precaudal plate is a derivative from the shield, from the organizer of the animal at the beginning of gastrulation, and goes up during the epiboly of the fish, goes forward like that, and induces the overlying ectoderm to be neural or not to be neural. And so it progresses like that, and at the end of the migration of the precaudal plate, when it reaches the tip of the head of the future animal, the precaudal plate is divided into two parts, the polster, that is the round part, and the more posterior part of the precaudal plate. So the polster is going to give rise to the hatching gland, and going to secrete these enzymes that are going to help the fish to hatch. And the posterior precaudal plate is going to the cells. We don't know exactly the fate of the cells. They are going more or less to disappear, or maybe to give rise to some derivatives on the head, we don't know. And so the difference between cavefish and surface fish is the spatial temporal, the dynamics of the MP4 expression in these two parts of the precaudal plate, the polster and the most posterior part. In surface fish, it's still expressed in the polster. In cavefish, it's kind of late. Or an alternative possibility is that it is turned off earlier in the polster and stays in the posterior part of the precaudal plate. So it's not a question of qPCR experiments have shown that the level of the MP4 expression is the same in cave and surface. It's just that it's not exactly in the same part of this precaudal plate.
And when you look at the sequences, what have you observed?
The sequences are
Is there variation within the coded region or different regions?
that we don't know. No. The coding regions identical for all these morphogenes. I mean, it's so important. It is so important these genes that if you modify the coding sequence, you're going to have big defects. So usually there is really a conservation of the sequence on all these type of genes. So the coding sequences are identical. The regulatory sequences, it is actually the aim of a project that we are actually trying to start and to get funded. Yes.
And one more thing. So you said that the enhance of the olfactory system is a product of the loss of the vision, the visual system.
It's not a product of the loss of vision.
Or
The
you said,
developmental evolution of the size, at least, of the size and of probably of the neuronal composition of the olfactory reticle. It's not a product. It's a trade-off. It's depending on these signaling systems, a bit more of SOMI here, a bit less of BLP4 there, a bit more of FGF here. This signaling system, they organize really the development of the entire part of the head of the animal, of the entire head. I just showed you placodes, but I could have done almost the same talk on the brain and on the hypothalamus versus the eye, the retina part of the eye. It's almost the same thing. So you favorize one part of tissue versus the other. It's really a sort of trade-off, and actually it's very interesting because it could be that this happens in all the various species that exist, and that could explain what we call the sensory specialization of animals according to lifestyles or habitats. Nocturnal animals, they have a slow vision, but good hearing and good olfaction, and good or they have sensory systems that are somatosensory systems that are very well developed also. So this could explain really the things that happen in the wild in every day. It's not a consequence. It's the origin is really early developmental origin.
Thank you for the talk. And I just wonder if there's such a selective force that are driving the development in the olfactory epithelium. But what I really want to know is if you have compared if there are such differences between male and female in cavefish
No.
for selecting
No idea. It's in for the embryonic, for all the developmental biology part, of course, it's impossible to know whether this is going to be a male or a female. For the larval, all the larval experiments that we did on 1 month old, all the behavior is done on 1 month old, 1 month old juveniles. You cannot know, it's too early to know the sex. And the only experiments that we have done on adults were done in those experiments that we did in the cave, in Seminarios, the field experiments, and we did not, I must say that we did not record the sex of the animals.
And comparing subterranean with Pachon, for example, did you notice some differences in the bacteria? So you know both of them are different ages, right?
Different. Both of them are larger than surface fish.
No, subterranean it's a lineage different from Pachon, so it's a new one and the old one, you know.
I was talking about the lineages, the lineage from Micos.
The stocks.
The stocks, okay. So what is the question related to the old and new lineage?
If there is a difference between the length and the development in the olfactory bacteria according to the lineage of the fish.
No, I have no clue. We don't have embryos of subterranean. I cannot know that. Or we could, and actually we did not sample fish. With the fish, we took them in the cave, we put them in the little swimming pool to do the behavioral test, and then we put them back in the water in the cave. We did not take them to do any histological things or measurements. I don't know that.
Okay, I think we are done. If you have more questions, you can talk directly with Doctor. We have a couple minutes.